Dear students, welcome again to our program of biology for 3rd secondary. We are going to complete our chapter that is talking about nucleic acid and protein synthesis. Let's go. The three main types of RNA that participate in protein synthesis, and these are: number 1, messenger RNA that is symbolized by mRNA. Number 2, ribosomal RNA that is symbolized by rRNA. Number 3, transfer RNA that is symbolized by tRNA. And we are going to know each one alone. Number 1, messenger RNA. First, you should know the DNA transcription into RNA. The transcription of DNA into RNA begins when the enzyme RNA polymerase binds to a sequence of nucleotides on the DNA that's called promoter. So, promoters are a sequence on DNA direct the RNA polymerase to the sequence of nucleotides on DNA to form mRNA or messenger RNA. Next, the two strands of DNA are separated and one strand only serves as a template for the formation of a complementary strand of RNA. After this, RNA polymerase moves along the DNA and joins the complementary nucleotides to the growing RNA strand one by one. The enzyme works only in the 3 to 5 direction on its DNA template, assembling RNA in the 5 to 3 direction. This figure represents the transcription of mRNA from the strand, the only strand that acts as a template of DNA. There is difference between the prokaryotes and eukaryotes in RNA transcription. There is a difference between them. In prokaryotes, a single RNA polymerase transcribes RNA, mRNA, and tRNA. But eukaryotes, there are 3 different kinds of polymerase. The messenger RNA of prokaryotes is ready for translation as soon as it's transcribed. But in eukaryotes, on the other hand, the original mRNA transcribed must be processed further in the nucleus before the mature mRNA is ready to enter the cytoplasm and participate in protein synthesis. At the beginning of each mRNA is a ribosome binding site. The ribosome binding site is a sequence of nucleotides that bind to a ribosome in such a way that the first codon AUG is positioned correctly for translation, and the last codon is called stop codon, which may be one of the 3 codons UAA, UAG, and UGA. At the other end of mRNA molecule, there is a polyadenine tail that composed of 200 adenine residues or nucleotides. It appears that this tail protects messenger RNA from breakdown by enzymes in the cytoplasm. This diagram illustrates the structure of messenger RNA. This is the ribosome site position, and AUG, this is the start codon, and there are 3 codons. They are called stop codon, and this is the polyadenine tail that prevent the breaking down of messenger RNA from any enzyme in the cytoplasm of the cell. Example number 2, or type number 2 of the RNA is called ribosomal RNA, that is symbolized by rRNA. Ribosomes are the organelles of protein synthesis. Do you remember what are ribosomes? From the inner structure of the cell, it's organelle inside the cell and help in the protein synthesis. That is consists of several types of RNA and about 70 kinds of polypeptides. In eukaryotes, the production of ribosomes takes place in an area of the nucleus called the nucleus, where several hundred thousand of ribosomes are produced per hour. Such rapid production is possible only because an eukaryotic cell's DNA contains up to 600 copies of the ribosomal. In eukaryotes, the polypeptides of ribosomes are made in the cytoplasm and then pass through the nuclear envelope. After this, enter the nucleus where the rRNA and polypeptides are assembled into ribosomal subunits. During protein synthesis, interaction occurs between messenger RNA and ribosomal RNA. The functional ribosome consists of 2 subunits, large and one small, one large and one small. And not engaged in protein synthesis, the subunits separate and move around independently. Each may join to the different subunits of the opposite type the next time they participate in protein synthesis. This structure illustrates the ribosome, that is consists of small subunit and large subunit. And the small subunit bind to the sequence of messenger RNA and help in protein synthesis. The third type of RNA is transfer RNA. The third kind of RNA that participates in protein synthesis is transfer RNA, which carries amino acids to the ribosomes. For each kind of amino acid, there is a specific kind of tRNA molecule that will recognize and transport it. Amino acids for which there are several different codons have more than one distinct kind of tRNA. Transfer RNA is transcribed from tRNA genes, often arranged in clusters of 7 or 8 transfer RNA genes in the same part of DNA molecule. All transfer RNA molecules have the same general shape. Parts of the molecule fold back in characteristic loops, which are held in the shape by base bearing between different areas of the molecule. This figure illustrates the structure of transfer RNA that has 2 sites. Number 1 is called amino acid attachment site, and this is called anti-codon site. So again, there are 2 sites of tRNA molecule which are important in protein synthesis. The first site is where the amino acid is attached to the molecule. This site consists of the 3 bases CCA, at the three end of the molecule. The other site is the anticodon site, which base pairs with the appropriate messenger RNA codon at the mRNA ribosome complex. This temporarily binds the transfer RNA to the messenger RNA, allowing the amino acid carried by the transfer tRNA to be incorporated into the polypeptide in its proper place. This figure illustrates the three types of RNA. Number 1, messenger RNA. Number 2, ribosomal RNA. Number 3, transfer RNA. So, at the end of this part, you should know RNA is very important substance inside your body that help your body to form protein. And there are three types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. The three participate to form a protein molecule. We have another topic today. We are going to talk about the genetic code. What's the meaning of genetic code? We are going to know. The genetic code represented by a particular sequence of nucleotides in DNA that is transcribed to a complementary sequence in triplet on mRNA. What's the meaning of this? The triplet mRNA goes to the ribosome where it's translated into a particular sequence of amino acids in a polypeptide which makes a particular protein. This shape illustrates what the meaning of genetic code. A sequence of nucleotides are transcribed into triplets messenger RNA, and each one is translated to a specific amino acid. We have a question here. Do you know what is the number of nucleotides responsible for selecting tRNA molecules which carry each amino acid? We said before we have 20 different amino acids are known to take part in the synthesis of protein. These are the 20 amino acids that help in synthesis of protein. But at the same time, there are only 4 nucleotides involved in the structure of both DNA and RNA. This figure illustrates the structure of DNA and RNA. Do you remember? DNA consists of 2 double strands of sugar phosphate backbone, and the nitrogenous base bind with another nitrogenous base with the hydrogen bond. But in RNA, there is 1 strand only, and these are the nitrogenous bases of DNA, and these are the nitrogenous bases of RNA. So, we have something new, is called the genetic language. Must have 4 letters. These 4 nucleotides or letters must somehow make up words, each one which stands for a particular amino acids. And there must be at least 20 different genetic codes to specify 20 amino acids. But we have only 4 nucleotides. So what is the solution of this problem? The words cannot be only 1 letter. If we consider that the genetic code is single or has one letter, so each nucleotide represents an amino acid code. So the words cannot be only one letter long, because in that case there would be only 4 possibles. If we consider that the genetic code is single or one letter, so each nucleotide represents an amino acid code. So the words cannot be only one letter long, because in that case there would be only 4 possibles. We have only 4 nucleotides, A, C, G, and U, and proteins could contain only 4 different amino acids. So the words cannot be just combination of 2 nucleotides long, because 4 letters arranged in all possible combination of 2 gives only 4/2 equals 16. Equals 16 different code words, still not enough to specify 20 different amino acids. We have another consider, that triple arrangement, or the codes or nucleotides are arrangement in 3 letters. The 4 nucleotides arranged in triplets, however, produce 4/3 equals 64 different codes words. That's more than enough to produce a unique code word for each amino acid. So the smallest theoretical size for a code word in DNA is 3, therefore 3 nucleotides. By 1960, there was considerable evidence of a triplet code. By deciding which sequence of 3 nucleotides coded for which amino acid called codon appeared very difficult. This table represents the genetic code of protein. As you see, one amino acid has 2 triplet codon. It should be noticed that the codons shown in the table are code words found in RNA, but the DNA code triplets are the complements of those shown in the table. And there is more than one codon for most of the amino acids. Also, there is codon for the start of protein synthesis, that is called AUG, and 3 stop codons. They are called UAG, UAA, and UAG, at which protein synthesis mechanism stops and gives signal. The genetic code is the point of termination of polypeptide chain is universal or general. Do you know why? Because the same codons code for the same amino acids in all living organisms like viruses, bacteria, plants, and animals, and fungi that have been examined. And this is a strong evidence that all living organisms on Earth now have been originated from common ancestors. Viruses and bacteria and fungi and plants and animals, all living organisms, their codons have been examined and they have the same codons of the amino acids. So, this is the time to know the steps of protein synthesis. Steps of protein synthesis. Number 1, protein synthesis starts when the messenger RNA binds to a small ribosomal subunit. Do you remember? In the ribosome site, binding site. This codon AUG is positioned correctly for the initiation of protein synthesis. The AUG codon then base pairs with the anticodon of transfer RNA carrying methionine. Then this methionine eventually becomes the first amino acid in the polypeptide chain. Now, a large ribosomal subunit binds to the complex and the reaction of protein synthesis itself can begin. And this figure illustrates the steps of protein synthesis. A messenger RNA, and this is a small subunit of ribosome. The small subunit of ribosome binds to the messenger RNA, and the AUG start codon bind to it. Then the transfer RNA carry amino acids to the large ribosomal unit. Then the protein synthesis began and form polypeptide of protein. We said before the ribosome has two sites where transfer RNA can bind the initiation codon AUG on the messenger RNA molecule. Is positioned as the first of these sites on the ribosome peptide P site. The messenger RNA codon for the second amino acid is lined up with the second side, the amino acyl site. The messenger RNA codon for the second amino acid is lined up with the second side that is called amino acyl site. From this point, the polypeptide chain elongates by a cycle of 3 steps. So, we are going to know the steps of polypeptide chain elongation. This structure illustrates the elongation of polypeptide chain. Number 1, codon regonization. Number 2, this is the step. This is the step of peptide bond formation. And then translocation. Then ribosome ready for next amino acyl transfer tRNA. Then amino end of polypeptide. The first step is the binding of next transfer RNA to anticodon complementary to the next messenger RNA codon. Number 2, the amino acid carried by the transfer RNA will be the next amino acid in the polypeptide chain. Second step is the peptidyl transferase reaction, which results in the peptide bond formation. The peptide transferase enzyme, which catalyzes the reaction, is an integral part of the large ribosomal unit. This enzyme attaches the first amino acid to second one by peptide bond, so that the first tRNA is now empty and the second is holding both amino acids. The third step in the cycle moves the ribosome along messenger RNA This brings the next codon to the ribosome's B site, and the cycle starts over again as the anticodon of the appropriate transfer tRNA bind to the codon, bringing the third amino acid into position at the A site. The growing polypeptide chain is attached to the newly arrived amino acid on this third tRNA, and the sequence repeats. Protein synthesis stops when the ribosome reaches a stop codon on the messenger RNA. Special protein called releasing factor binds. Special protein called a releasing factor binds to the stop codon and causes the messenger RNA to leave the ribosome. The ribosomal subunits separates as the 5' end of the messenger RNA emerges from the ribosome. It may bind to another smaller ribosomal subunit, which initiates protein synthesis again. One messenger RNA attached with 100 ribosomes to form a cluster called a polyribosome. One messenger RNA attached with 100 ribosomes to form a cluster called polyribosome. One messenger RNA attached with 100 ribosomes to form a cluster called polyribosome or polysome. And this figure illustrates the steps of protein synthesis. A messenger RNA, and this is a small subunit of ribosome. The small subunit of ribosome bind to the messenger RNA, and the AUG start codon bind to it. Then the transfer RNA carry amino acids to the large ribosomal unit. Then the protein synthesis began and form polypeptide of protein. This structure illustrates the elongation of polypeptide chain. Number 1, codon regonization. Number 2, this is the step of peptide bond formation. And then translocation. Then ribosome ready for next amino acid transfer tRNA. Then amino end of polypeptide. I hope you understood our lesson today and study well, then apply your studying by answer some exercises. Thank you for your listening and goodbye.